Good morning, everybody. I had a dream, and the dream was to organize a workshop like this. This was made possible because I had the board of the Norwegian Society for Immunology supporting me, and not the least because Matzinger accepted my invitation to come to Oslo. This is a workshop which I will come back to the format in a second. I just forgot to mention, to thank the Norwegian Society for the board of the NSI is the organizing committee. The treasurer of the NSI is paying for most of the workshop, but not everything. So we do have some sponsors. Our gold sponsor is the Scandinavian Journal of Immunology, our official journal. So remember to submit papers to the Scandinavian Journal. We also have two other silver sponsors, the Center for Immune Regulation and RearNet, and patrons of the NSI who are supporting us the whole year. So this is a workshop, which means, so for a workshop to be successful, there are two important things. First, it should be small, and there should be a lot of discussion. So we obviously failed on the first point, but we just asked for it when we invited Polly. You cannot make it small if Polly is the main speaker. But the discussion, we made sure that there should be a lot of discussions. As you can see on the, and this is really important to make it successful. So as you can see in the program, there's lots of discussion time between talks. However, you don't need to wait for the discussion time to ask questions. Just raise your hands. I think we have a microphone or two. So it should be really interactive. On the top of that, and that was a request from Polly when she accepted the invitation, is that we should be flexible on the schedule. I know from previous experience with Polly talking that she never gets enough time for talking. But that's because in most conferences she typically have 1 hour off. So to make sure that this will not be a problem, we have a 2 days workshop. It will be biased in that sense that Polly will have much more time than anyone else to talk. That's fine. Polly has also the reputation to take over the whole discussion. And my strategy there was, again, to have a 2 days workshop so that she will have time to say everything she wants to say, and there will be still some time left for others. Should work, doesn't it? And the final requirement by Polly was flexible schedule and also possibility for evening sessions. So we'll actually keep the discussion going during tonight. It will be pizza and beers and dinner tomorrow. So no limit in the discussion. Now, to make the workshop even better, I not only invited Polly, but I'm very happy to have two other foreign guests, and not the least, Peter Brecher and Colin Anderson. They represent different views, so the workshop is on the danger model, but I think it's very important to have different views expressed to make a better discussion. Yes, so let's start. The first speaker, and I think she will be the last speaker as well, of course, is Polly Matzinger. She's known for many things. I don't think I will need to introduce her much. She has been a Playboy bunny. She's known for that. I checked at home my old issues of Playboy magazine to find the original article on the danger model. But someone told me that she was a Playboy bunny in a club, wasn't it? Anyway, she has done much more than that. She changed career, started doing science. And it's just great to have you here, Polly. The floor is yours.
Thank you. This is not the beginning of a striptease. It's just hot in here. Yeah, I noticed that the last time there was a meeting where Alex and Colin and Peter all discussed the danger model, I wasn't invited. So this is my turn. I also noticed that on the program, is this thing too loud? It feels a bit loud to me. How about the back row? Okay? It's okay? Okay. I noticed there was nobody talking about allergy in the context of the danger model or in the context of anything else. And so I was supposed to talk about danger, transplants, fetuses, and tumors, and I'm going to add allergy and maybe talk a little bit less about transplants because we're going to do that, I think, when Colin speaks. Okay, so this is a workshop on the danger model. I presume that you all know the basic assumptions of the danger model. Is there anybody who feels that they don't? Okay, good. So the basic assumption, and by the way, there's a detail here that's important that I don't usually have an opportunity to express because I so seldom get to talk to an audience that really understands the basic assumptions. The basic assumption is that you don't get a primary immune response unless you activate an antigen-presenting cell, and that you activate an antigen-presenting cell usually when something is doing damage. Right? Now, there's an important word in that assumption, and that is primary. Because if we have time, I can get into how I think that secondary immune responses don't necessarily need damage to activate antigen-presenting cells. So if I don't get to that, will somebody please remind me about it later? Okay? So people tell me they teach danger in their immunity classes, and so I ask them what they teach, and they tell me that in the section on antigen presentation and so on, they talk about how there are endogenous alarm signals that activate antigen-presenting cells. And that's the danger they teach. And Mel Cohn, who is my favorite immunological theoretician, I still think he's the best living theoretician on the planet for immunology, would say that that is cutting off too small a slice of the pie. So the slice that people are teaching is that there are endogenous alarm signals that activate presenting cells. But there's the whole rest of the pie of immunology. And what I find is they don't then apply that to other stuff. So for example, I ask them, what do you teach about why mothers don't reject their fetuses? And they tell me, you know, what's in the books. Fetuses are immunosuppressive, and there's a barrier between mother and fetus. How many of you have learned that? There's a barrier, the placenta is a barrier, both physical and immunological barrier between mother and fetus. Okay, that's not teaching the danger model. What they're teaching is a tiny portion. So let's do mothers and fetuses, right? For fun, yeah? How does the danger model deal with that, and how do other models deal with that? And what is being taught about mothers and fetuses is still what Medawar wrote in the early 1950s. It's a self-non-self explanation for mothers and why they don't reject fetuses. What did Medawar write? He wrote two important things, both of them evolutionarily stupid, and both of them incorrect. So the first one is that the placenta is a physical and immunological barrier between mother and fetus. Right? Is this still written in textbooks? and we know it's wrong, right? The idea is that a few antibodies can get through, but the cells that could reject the fetus can't go across the placenta. Well, that's wrong, right? We know that there's traffic in both directions. Right? So we know that fetal cells go into the mother, and there are people isolating those cells from the blood of pregnant women using cell sorting, and then using them to do a non-invasive prenatal diagnosis. Right? This is becoming more and more common in the clinics. So there's traffic of fetal cells into the mother. There's also traffic of maternal cells into the fetus. So this was first discovered at Eurotransplant in Leiden in the Netherlands by Jan van Root and his group. There they have people waiting for kidneys. They need a kidney transplant. They're on dialysis. And sometimes they have a living donor willing to give their kidney. When you have a living donor, you always test the immune response of the recipient, the potential recipient, against the potential donor first. Because if it's already a strong response, there's no sense in doing that transplant because it won't last longer than a week. So mothers are often donors. And it turns out that 25% of kidney patients make no immune response against their mothers. So you know that they inherit HLA antigens from their father, they inherit HLA antigens from their mother. They don't respond to the non-inherited HLA of the mother, even though they do respond against the non-inherited HLA of their father. And Lee Zhang and Rick Miller followed that up in Toronto with mice, where you can label the father, the mother, and the F1 progeny. And they discovered that 25% of mice are carrying maternal hematopoietic cells. So this says there's traffic from mother to fetus. 50% of human skids, so these are the kids that have to live in a bubble, have T cells from their mothers. So there's traffic from fetus to mother, and there's traffic from mother to fetus, and yet the mother doesn't reject the fetus. Why? So the danger model says it's because fetuses don't look dangerous. The kind of death that goes on in fetal life is programmed cell death. Right? You want to make a hand out of that primitive paw, you kill the cells in between, you kill them by programmed cell death, they're scavenged by their neighbors, they don't release alarm signals, and so you don't get an immune response. It doesn't matter that the fetus is foreign. Without the alarm signals, you don't get an immune response. So should the fetus get infected, you do get an immune response. And if in the process of clearing the infection you clear the fetus, at least you're alive to have another one. Right? And we know the clinical literature, both veterinary literature and human medical literature, knows that spontaneous abortion is a common concomitant of infection during pregnancy. So that was wrong, Medawar's idea. So his second idea was also kind of evolutionarily dumb, and that is that fetuses are immunosuppressive. That's great. Now you take the individuals that are carrying the next generation and you turn them into AIDS patients. It's an evolutionarily unbelievably dumb idea. And it isn't true. How many of you have carried fetuses? All right, pick the closest one here. Did you have to live in a bubble? No. No. Has anybody noticed this? This is still written in textbooks. Pregnant individuals are amazingly healthy. Human, all those cows and sheep and pigs out there, they don't live in bubbles. Now, it's true that there's some parasites that have learned to take advantage of the pregnant state. And here's one story about that used to be thrown in my face as evidence that pregnant females are immunosuppressed. And it actually turns out to be evidence that they can make a primary immune response while pregnant. And that's malaria. So in malaria endemic areas, a woman who's going through her first pregnancy has a terrible time if she gets infected by malaria. She can die. Her fetus can die. If they don't die, there's low birth weight. And so this was evidence that pregnancy was an immunosuppressed state. It turns out not to be true. It turns out that there's a variant of malaria that takes advantage of pregnancy. So malaria-infected red cells can be mechanically removed by the spleen because they don't look like real red cells. Right? They're knobby. And so malaria has learned not to let itself get through the spleen. And it does that by putting on the surface of the red cell certain molecules that bind to the endothelium of blood vessels. And so the infected red cell will bind to the endothelium and stay there and not go through the spleen. Well, there's a variant of malaria that puts up a molecule called VAR2-CSA that binds a chondroitin sulfate that is only found on the placenta. And so this is a variant of malaria that can only grow in pregnant females. And so the pregnant female in Africa who's having her first pregnancy, who's usually 13 years old, has never seen this malaria variant before, and she's making a primary immune response. And as we all know, a primary immune response is slow and weak. And so what happens is they get sick during that first pregnancy. But by the second pregnancy, they're doing better. And by the third, they're fine, if they survive the first one. Okay? And Lars Høvid, who I don't know if he's Norwegian or Danish or one of those Scandinavian types, Lars Høvid has actually done a study where he went into malaria endemic areas in Africa and bled people and asked if they were making antibodies to this particular variant of malaria. And what he found was that every woman who's ever been pregnant is making antibodies to this variant, and almost no man is. Which is pretty good evidence that you can make a primary immune response during pregnancy. So toxoplasma is another parasite that can take advantage of the pregnant state. And I don't know enough about the details of toxo to tell you that story, but I bet you that if somebody went to look at it, they would find a story similar in some ways to the story of malaria. Women during pregnancy are not immunosuppressed. Right? It's a stupid idea and it's wrong. Actually, I have found in my career that if something seems to be evolutionarily dumb, it's probably not true. Okay. There are some people who have suggested that what the placenta does is to create local immunosuppression. That you're only immunosuppressed at the placenta. That's also pretty dumb. That means if your fetus gets infected, you can't do anything about it. And probably you and it are dead. Right? And it's also not true. And we're not going to discuss that one until the end of this meeting, where we talk about the kind of immune response that the immune system makes. Right? I don't have time for that one today, but maybe we can discuss it later. Okay, that's pregnancy. The danger model says you don't reject your fetus because it doesn't look dangerous. It is dangerous. Fetuses are dangerous. They suck calcium out of your bones, and they're actually pretty selfish. They're a parasite, just like malaria, fighting for the same eternal resource, which is blood. And under starvation conditions, it's the very young, the very old, and the pregnant who die first. Because the fetus doesn't care. It just wants that nutrition. Okay? So it's dangerous, but it doesn't look immunologically dangerous. And that's an important point. Tumors are dangerous, we'll get to that, but they don't look immunologically dangerous. Fetuses are dangerous, but they don't look immunologically dangerous because the death that goes on in fetal life is programmed cell death. Okay, so you don't reject a fetus because it doesn't look dangerous. Why do you reject a transplant? And by the way, please start this discussion. Don't just sit there and wait to the end. If you know something that doesn't fit, bring it up. So the question is, I don't know if the people watching worldwide can hear the question, so I'm going to try and repeat them. I think every speaker should do that. So why does the placenta lack HLA molecules that are important for rejection? So that happens to be true in humans and not in mice. And so my guess is there's another reason for it in humans. Red cells don't express much HLA molecules either, and that's true in humans, but not in chickens. So I think if we want to have generalizable ideas about immunity, we should have those things that apply to all kinds of species. And I would guess if we went to look a little more carefully, we would find that there was another reason for that. And one reason might be Klaus Schar's kind of missing self idea. There are NK cells that seem to be important, not for killing at the placenta, but for helping the placenta to attach. And perhaps it's important that the placenta therefore not have those HLA molecules because they turn NK cells off. I bet there's at least one person in this audience who's thinking, why is it that Rh negative mothers reject Rh positive fetuses? Anybody? No? Okay. So the question is, it's not a question, it's a comment, that it's commonly known that women who have autoimmune diseases often feel better, and allergies, often feel better during pregnancy. And you know, it's really interesting, and it's true for some autoimmune diseases and not others. And can you hang on to that one until we do autoimmunity? Well, okay, let's just do one example of it. Take lupus erythematosus, right? Systemic lupus. There are forms of lupus where the woman gets better during pregnancy, and there are forms of lupus where she gets lupus during pregnancy and not after. Right? So in some cases it gets worse, or even appears during pregnancy, and in other cases it gets better during pregnancy. And I'm going to discuss that during the autoimmune session. Yeah? And if we have time during this one, we can come back to it. Let's do Rh just quickly. Rh negative mothers make immune responses to Rh positive babies, and people say, well, where's the danger? So God is in the details. The detail that's important here is that this is not a problem in the first pregnancy. It's a problem in the second. Women get primed at birth. Right? Birth is dangerous. Right? You have ripping and bleeding, surgeons with knives, cutting. This is not programmed cell death. This is when they get primed. And physicians know this because they don't worry about this discrepancy in the first pregnancy. They give anti-Rh antibody to a woman just before she gives birth to cover up those antigens and not let her immune system see them. Birth is dangerous. the details matter. I saw another hand. So what is my opinion on surrogate mothers? What's going to be the immune response in a surrogate mother as opposed to a normal mother? Personally, I don't think it's going to be different. So my prediction, and it could be wrong, right, is that a surrogate mother would not have a problem with a second pregnancy, that a normal mother wouldn't either. Because when you think about it, the genetic differences that occur with a normal mother, right? That's at most one HLA difference, right? Plus a whole bunch of minor histocompatibility differences. It's only twofold for a surrogate mother, right? She's got maybe 2 HLAs to worry about. So I don't think it's going to be a huge difference. Yeah, I could be wrong. Right? Hi. Joanna. At birth.
While giving birth. But there is a fraction who actually are immunized because you have readings from the fetus to the mother prior to birth.
Yeah, very good. Very good point. So you just explained it. Does anybody understand this? Does anybody?
Well, you say that the bleeding is the danger.
Yeah, I do. So I had an obstetrician for a while who was working in my lab, Liz Bonney, and she told me that there are women who have what are called fragile placentas. There's a fraction of women that have a fragile placenta. And what happens with these women is that the little fingers of fetal tissue that sit in the lakes of maternal blood sometimes break off. That is not programmed cell death. So I would predict that if there were, keep my English straight, if there were a RH difference under those conditions, that those women would start to make the antibodies towards the end of the first pregnancy. It's amazing how many things the danger model can keep explaining. I mean, there's some it can't, but it's pretty good. You just keep going, right? Okay. Yes.
Take the embryo from one species, put it in the kind of surrogate mother of another species, and get it done. Why? So
why doesn't that work more often?
Because it's no danger anyway. I mean.
So the question is, why is it so difficult to take an embryo from one species and stick it into a pseudo-pregnant female of another species and have it work? I don't think that's an immunological problem. I think that's, they have different chromosomes, they have different instructions. I mean, the fetus and the mother have to be talking to each other all the time. So I would be kind of surprised if you could do this across all these different species. There may be species that are close enough that you can do it. For example, you can take cow embryos and put them into rabbits. It's kind of dangerous for the rabbit, but you only leave them in the rabbit for a short period of time, and then you ship the rabbit to another country, and then they take those cow embryos out and put them back into cows. That works. But it only works because the cow embryo goes a couple of weeks before it implants. And so you're taking these embryos when they still have a zona pellucida, and you just stick them into a kind of nice, warm, nutritious environment, and then you take them out again. But they probably wouldn't implant very well. I don't think that's an immunology problem, except maybe the NK cells.
May I have a question again about this RH immunization? If the mother is immunized...
Slow down, because you're in the back and there's an echo, so...
If the mother is immunized during birth of the first child against RH, why doesn't she immunize against the other HLA molecules of that child? So why the second pregnancy, why only RH is problem in the second pregnancy and not the other?
Okay, that's a really good question. Why, if the mother is getting immunized to RH in the first pregnancy, and it's still a problem for the second pregnancy, isn't the immunization that you would expect her to get against the HLA antigens also a problem in the second? Yes? Okay. So here's my hand-waving explanation. So people sometimes ask me, don't my wrists get tired of hand-waving? Here it is. Right? The RH problem is a problem of antibody. Right? So you get the antibodies against RH, and those antibodies last a very long time. Now, I don't know why they last a very long time. It could be that they're cross-reactive environmental antigens that maintain them. I don't know. Yeah, that I don't know. But they last a long time. The activity of a cell that's going to reject a graft is usually an activated CD8 effector or an activated CD4 making nasty cytokines. That activation state doesn't last so long. Those cells go back into a resting memory state. And they need to be reactivated. So, for example, when you vaccinate someone, right, you add adjuvant that does damage, et cetera, activates APCs. You get a bunch of activated cells. They stay activated for a while. Most of them die. And the rest go into a resting memory state from which they have to be reactivated. Now, that's fine for a vaccine because you prime, you increase their frequency. You boost, you increase their frequency again. Usually that's about as good as it gets because there are homeostatic mechanisms that keep it there. But they're in a resting memory state. When the pathogen that you vaccinated against comes back, it does the damage that reinitiates that response and takes the APCs to a state where they can reactivate the memory cells. Now you've got memory cells, resting memory cells, and you've got a fetus that is not sending alarm signals. So it doesn't matter that you have resting memory cells. Does that make sense?
Yes, but I would think that to make antibodies, you need T-cell help. So you need kind of a length of activation.
You need T-cell help. It's true. It's true, you need T-cell help to make antibody. But then we know that you don't need T-cell help for those B cells, some of them, to continue to make antibody. For example, there are B cells, they're called long-lived B cells that live in the bone marrow and just keep making antibody. I don't think anybody's ever looked to see if you remove all the T help, if those B cells continue to make antibody, but my bet is they will. They no longer have MHC class II on their surface, so they're no longer bindable, if you like, by a helper cell. They're just there in the bone marrow as antibody factories. Now, Peter might disagree. I don't know, do you disagree that long-lived bone marrow cells don't need help?
I don't know.
Yeah, I don't know either, actually, to be fair. And so maybe this is an experiment worth doing, to ask, or to go into the literature and discover that it was done in the 1960s. It's amazing what was done that long ago. But that would be my explanation for that.
Now, on the other hand, polysaccharide antigens, for example, you don't need T-cell help to stimulate B cells. Polysaccharide antigen can cross-link all receptors in B cells. So, in that case,
So there are polysaccharide antigens to which you don't need T-cell help. I don't know that it's just the cross-linking by the polysaccharides that do it. It could be that those polysaccharides are also on bacteria that have other TLR ligands. So I don't know. If you put a polysaccharide antigen on a red cell by itself, whether you would get a response in the absence of CD4s, I doubt it. You don't. Okay, there you go. You don't. You need something besides just the cross-linking of the polysaccharides. God, a supportive statement from Colin Anderson. That's amazing. Okay, shall we leave fetuses? Are we done with fetuses? Oh, my God. Okay, we're going to do transplants later, so I'm running out of time. So what we're going to do is we're going to skip transplants, sorta, for now. No, actually, I'm going to do 2 experiments that Colin did. The prediction from the danger model is that if you could do a transplant without doing damage, it would not get rejected. Right? So here's the beginning of a controversy at this meeting. My prediction is that if you could do a transplant without doing damage, it would not get rejected. If you could do a transplant and block co-stimulation, it would not get rejected. If you could do a transplant and upregulate co-inhibition, it would not get rejected. So, Colin did an experiment a long time ago, and they discussed this at the last meeting where I wasn't invited, where he took immunodeficient animals, rag knockout animals, and transplanted them under the kidney capsule with neonatal hearts. And these hearts differed by about 20 to 25 minor histocompatibility antigens. And had those mice had an adaptive immune system, they would have rejected the hearts in anywhere from 14 to 30 days or so. But they don't have an adaptive immune system, so they don't reject the grafts. And he allowed the grafts to heal for 9 months or 1 month. And then he gave the mice an immune system by giving them fetal liver cells. And he asked the question, if a transplant has been sitting in the periphery, healing for 9 months, and therefore might be in a state where it is no longer sending alarm signals, would this new immune system reject it? And the answer was no, they were not rejected. But if they had only been there 1 month, they were rejected. Okay. And he then tested a second part of the danger model, which suggests that if a naive T cell that leaves the thymus specific for an antigen in the periphery meets that antigen in the periphery under conditions of health and not damage, that T cell will die. Yeah. And the way he tested that was to immunize these mice, let them sit for 5 months, so that now the tissue not sending alarm signals is interacting with the immune system. T cells that see it should be able to circulate and run into it in a healthy state and die, and therefore this animal should become tolerant. If it becomes tolerant, you would not be able to immunize it. And so he immunized them. And in his first experiment, which was done when he was still in my lab, I'm going to show you the very early data. And by the way, people believe these data if I draw them on a computer. But if I draw them on a blackboard or a piece of paper, they don't. So you can draw them on the computer as well. Here are the data. This is percent kill. And this is a titration of killer cells. And these were the control mice. They never had a heart. So these mice sat in the same cages for the same amount of time. They sat for 9 months. They got an immune system. They sat for another 5 months, and then they were immunized. And he tested for killing against the donor. And there were 2 that did phenomenal killing, 1 that did very good killing, and they didn't kill self. You can see why it doesn't matter, right? You've got lines here and lines here. Does it matter that you draw them on a computer? Okay, these were the mice that had their hearts for 1 month. They rejected, and they also made killers. Not as good as these, but still made killers, and they didn't kill self. And these were the mice that had had their hearts for 9 months, and they made no killers against either the donor or self. Now, what the conclusion from those experiments was, if you put the graft in and you leave it there long enough, under healthy conditions, it will not induce rejection. And if you let it interact with the immune system for a while, it will induce tolerance. Now, Colin followed these experiments up, and he did another experiment where he did basically the same protocol, except this time, instead of using normal hearts from a normal mouse, he used hearts from a mouse that had no T cells. Well, no B cells either. And he got a slightly different result. Only slightly. Here you're looking at percent rejection. Right? And what he got was just like in the other case, if the heart sat for 9 months and they came from a mouse that had T cells, there was no rejection. If they had sat for 9 months and they came from a mouse with no T cells, 50% of them rejected. And 50% didn't. And Collins' conclusion from that, which I think is correct, is that the T cells that were living in the original graft went to the thymus of that animal. And when you gave that animal back an immune system, they induced tolerance. Right? So when you have lymphocytes in a graft, and you can put the graft into an immunodeficient recipient, the immune cells that live in that graft, very small number, can go to the thymus, and then when this recipient gets an immune system, the T cells growing up in that thymus will meet these donor T cells and be deleted. And that's a very efficient way of inducing tolerance. However, it's not the only way, because 50% of the animals that had grafts that did not have those kinds of migratory T cells also accepted their grafts. And in fact, when he regrafted them, 30% of them didn't reject the second graft. So what I think is going on here is that you have two forms of tolerance, or two places where you can tolerize T cells. You can tolerize them when they're growing up in the thymus to anything that is in the thymus. And it's very difficult to tolerize them to things outside the thymus that way. Right? But you can. And you can if you give them enough time. And I would argue that the 50% here would have gone to 90 if he had left them longer to heal. So here's another thought. One of the things that I thought from these experiments a long time ago is that a tissue doesn't fully heal until it becomes innervated. And innervation takes a long time. And this comes from work from Walter Land, who was the head of surgery in Munich, head of experimental surgery in Munich, that if you look at heat shock proteins and those kinds of things in a graft from a person who has just died, that tissue already starts to act not very happy. It's no longer getting neural signals. And it starts to upregulate heat shock proteins and remodeling proteins and so on. So, transplantation tolerance. If we can get a tissue in and leave it to heal, it will not induce rejection. If we can get a tissue in that will also send cells to the thymus, that gets even better. Okay? We're going to have that discussion later, I presume? Okay. Let's, for fun, do allergy. How much time do I have left? Given that we're discussing while we do it? Somebody tell me how much time I have left. Alex. How much time do I have left?
5 minutes plus discussion.
Okay, well, we've already taken 15 for discussion, so... Okay, great. Someone do allergy and tumors? So let's do allergy. There are two questions in allergy, which a lot of people put together as one, but I try to divide them in two. The first one is what makes an allergen an allergen? So this is a question that was first asked by a guy named Gunnar Aas. And he said, the house dust mite has thousands of proteins, and 2 of them are allergens, DRP1 and DRP2. The cat has even more proteins, and yet there's one allergen, FeLD1. And his question is, what makes an allergen an allergen? Why are there some molecules that are allergenic and other molecules that are not? And the second question in allergy is, why do some people get allergic and other people not? Why do some people respond to these allergens and other people not? I think that's the wrong question. So I think I can answer the first one. What makes an allergen an allergen? So an allergen, in my book, is an immunogen. And what makes a molecule an immunogen? It's either causing damage, or it's packaged with something that causes damage, or it mimics an endogenous or exogenous alarm signal, or it's given to you at a time when you have damage for another reason. All right, so the first one is easy. DRP1 is a protease that attacks the surface of lung epithelium and also the surface of B cell. So 1, it's an allergen that causes damage. 2, something packaged with something that causes damage. Right? Bee venom is not innocuous. Right? There's a lipase in bee venom. There's all kinds of stuff in bee venom. You could imagine that anything that you find in bee venom could be an allergen because the venom itself is causing damage. All right, so that's packaged with something that's causing damage. With damage. Number 3, something that mimics an alarm signal. So I don't actually know of any allergen that fits in that category yet, but given the number of alarm signals, DNA, RNA, DNA, RNA, HR proteins, the breakdown products of hyaluron, uric acid, ATP, the list is getting longer and longer and longer. You could easily imagine that there is something out there that mimics that. And the 4th category is kind of fun, that you get it at the time of damage. And I think peanuts are here. So I didn't used to know why we have peanut allergy. Peanut allergy is an allergy that only started really coming up in the 1990s. And you realize that people in Indonesia have been eating peanuts for thousands of years. The Dutch have been eating peanuts ever since they took over Indonesia, and so on. We eat peanut butter, right? Peanut allergy started in the 1990s. What's going on with peanut allergy? So in Montreal, there's a guy, I can't remember his name now, who works on peanut allergy. And he told me that in Canada, they are banning peanut oil in baby creams. Now, why is peanut oil in baby creams? Because it's a great oil. So when you have a baby that has a rash and you're putting baby cream on him, you're smearing peanut oil into the skin of an organism that has damaged skin and therefore presumably activated antigen-presenting cells that are picking up that molecule and giving you an immune response. So the anecdote that kind of proves this idea is that at the NIH, there's a couple, a man and his wife, who are both immunologists. They study tuberculosis. And they had a baby that had a lot of rash. And they knew this idea about peanut oil. So they went to special organic stores and got baby cream that used sesame oil instead of peanut oil. And when that baby was 1 year and a half old, he anaphylaxed in his daycare when they fed him hummus, which has sesame oil in it. Right? So if we stopped rubbing our babies with peanut oil and gave them peanuts to eat instead, we could probably stop this rash, in fact, of peanut allergy. Okay? So an allergen is either something that causes damage, like DRP1. It's packaged with something that causes damage, like bee venom. And by the way, pollen is probably here too. Birch pollen, which in Finland is a terrible problem. Pollen, the little pollen particles, have proteases on their surface that they use to dig through the pistil of the plant to get to the egg at the bottom. They're covered in proteases. They're not innocuous. So, the reason an allergen is that it causes damage. It's packaged with something that causes damage, mimics an endogenous or exogenous alarm signal, or is given to you at the time of damage. Alex?
What about other food allergies, like nuts, shrimps? It doesn't look very dangerous.
Well, they don't look dangerous as food. And so nuts, peanuts don't look dangerous either, right? And so God is in the details. So the short answer is, I don't know. And the slightly longer answer is that I would look for one of those categories. Right? Maybe shrimp oil is used in something. I don't know. Right? But at least the peanut story tells us at least where we can start looking. Gunnar asked this question 40 years ago, and there has not been an answer since. So here's a potential answer coming from the danger model. Either it's dangerous, or it's packaged with something that's dangerous, or it mimics an alarm signal, or it's given to you at a time of danger. Right, so for those of you who can't hear, this woman's sister, I don't know your name, I can't read it this far, sister has contact dermatitis, right, has dermatitis, and also fish allergy. And so she's saying, well, maybe she was cooking fish when she had the skin rash, or maybe whatever she puts on that dermatitis has some fish product. You find fish products in all kinds of things. So we should start looking. Now, the second question in allergy is a question I can't answer, but I'm going to change the question, because I can answer that one. So, why do some people respond to these allergens and others don't? I think is the wrong question. I think we all respond to these allergens. It's just that most of us make IgA or IgG, and a few people make IgE. And I don't know why a few people make IgE, but I have some thoughts which will come at the end. And I've asked allergen people to do the following experiment, and I've been asking them to do this for 18 years. And it's incredibly difficult to get people to do a really simple experiment. Here's the experiment. Take some families that have a few kids, where some of the kids are allergic and some of the kids are not. And test the kids who are not allergic for their immune responses to the same allergens that the allergic kids are allergic to. And the prediction is that the non-allergic kids will be making IgG, and the allergic kids will be making IgE. They're all responding, right? Because the allergen is just an immunogen. But some of them make IgG and some of them make IgE. So, 2 years ago at the World Immune Regulation meeting in Davos, this postdoc came up to me and said she'd heard I'd been asking about this study, and they had done this study, and indeed, in their hands, many of the kids who are not allergic, in the same family as kids who are allergic, are making IgG. She can't get it published. It's really hard to get something published when people aren't ready to hear it.
So it's my understanding that Actis has done experiments where he successfully desensitizes in one individual. They go from predominant IgE production to IgA and IgG4 production. The other.
This is actually, it's not just actus.
No, I know it's not just that. But
Yeah, so,
saying that even doing it in one person, you can see that you can go from an allergic state to non-allergic. It seems to me it's very powerful evidence.
it's,
What a lot of people think is
so Peter thinks that it's great. I love it. All right. So here's a really good immunologist who has looked at other evidence, which is that when you take people who are allergic and you do the right things to them, they stop being allergic. But it's not because they're tolerant, even though they often call it tolerance, and he calls it tolerance, actus. It's not tolerance. What they've done is they've switched the response to a different class, which doesn't give you allergy. Slit therapy, sublingual immunotherapy, they take molecules that you're allergic to and put it under your tongue and make you eat it. And what's going to happen then is you're going to get an IgA response. They call it tolerance too. But it's really a switch from IgE to IgA. So that makes my prediction actually quite an easy prediction to make. Except that these people still think that the non-allergic people are not responding. Anyway, agreement from Peter. Okay, that's great. I love it. That's allergy and tumors. I'm going to do tumors very quickly. Why don't you reject a tumor? There are a lot of details here that maybe we can get into in the discussion, but I think the basic idea is that just like fetuses, tumors don't look dangerous. For a long time while a tumor is growing, not the one you inject, not the one that you induce with methylcalanthine, but the normal tumor that has mutated and has now lost growth control and is dividing or multiplying, since division and multiplication in biology are the same. Right? What's wrong with this tumor? It's too healthy. Right? It's lost growth control. And if it's expressing a tumor antigen in MHC on its surface and doesn't have, well, in fact, the tumor is still a tissue. The lung cancer is still a lung cell. We know it's a lung cancer and not a metastasizing colon cancer because the pathologist can see that it still looks like a lung cell. So this cell has a lot of the properties that the lung cell has. And one of the properties that a lung cell might have is that the balance of co-stimulatory molecules and co-inhibitory molecules is high for co-inhibition because cells don't want to induce immunity. So this is a tolerizing cell. And what's going to happen to a T cell that sees it is it's going to get what Melcone would call signal 1 without 2, and what Sinclair and Collin, and I agree with them, would call a balance of tolerance versus a balance of activation. It's going to tolerize the T cells that see it. So what happens while this tumor is growing is that it's killing off the T cells that can see it. Now, eventually, if it gets really big and it's not very well vascularized, it'll start to die in the middle at starvation death. And starvation death is programmed cell death. So even at that stage, it's not going to induce an immune response. It's only when this tumor gets really big and the amount of death that's going on overloads the local scavenging capacity that now you will get the release of alarm signals, the activation of presenting cells if they live there. And as these cells die, they're going to release antigens, and those antigens will be presented, the tumor antigen, by now an activated APC. And that T cell is going to get stimulated, and it's going to make a small army, and that army is going to go back to the tumor. And each of those killer cells, it is estimated, can kill 3 to 30 targets. I'm off the graph. What happened? Press 1. Press any. Voila. All right, so you have a presenting cell, presenting tumor, turning on T cells. The T cells make an army. Each one of the killers can kill 3 to 30 targets before it has to be re-stimulated. The tumor's been tolerizing for this whole time, so by the time you get to that stage, you've got 3 T cells left. Each one of them becomes 10. Each one of those can kill 30 targets. And they're going to go do that. They're going to go back to that tumor, maybe, we can talk about that, and kill targets. And what's the tumor doing in the meantime? It's making millions of cells more. You've lost the race. So, Steve Rosenberg, who's America's Mr. Cancer, had a really cool idea. He said, I'm going to take the TILs, the tumor-infiltrating lymphocytes, out of these tumors, and I'm going to grow them up in bathtubs full of IL-2 and put them back in. And they're going to kill the tumor. And if you heard him talk 15 years ago, you would see the same set of slides. He had a patient who had a melanoma metastasis in the lung that was the size of a softball. A softball is a baseball that they let women play with in America. It's a bit bigger and a bit softer. Right? So he took the TILs out. He grew the tumor-infiltrating lymphocytes. He grew them up in bathtubs of IL-2. He put them back in, and the tumor went down to the size of a baseball. And then he did it again, and the tumor went down to the size of a golf ball. And then he stopped. And the tumor grew out and killed the patient. So what's wrong with this picture? He had a treatment that worked. He had a treatment that makes the tumor get smaller, and he stopped doing it. Why did he stop doing it? Because he's in vaccine mode. Because he's stuck in the old model. This is really a place where what you think influences what you do. And if that's all you remember from this talk, that's good enough. What you think influences what you do. And what he thinks, and what a lot of people think, is that once you turn the immune system on to a tumor or to any other antigen, the mere presence of that antigen is sufficient to maintain the response. And that's not true. Right? Immune responses have immune kinetics. Right? We know this from all kinds of other studies. You do a response, it peaks at day 7. Killer response, it's gone by day 14. If you want that immune response to keep going, you need to do it again. And if you want it to keep going, you need to do it again. So what's happening if you have a tumor? You have a tumor that's growing, it's been growing. You do something, you get an immune response, the tumor gets smaller. If you've gotten rid of the last tumor cell, great. If not, you boost, and the tumor has been growing again, and it gets smaller. And then maybe you boost again. But in the meantime, as this immune response goes away, the tumor starts to grow again. And then you stop. And what happens when you stop? The antigen-presenting cells that you have activated with your treatment, like say it's a vaccine, live for 3 to 21 days. After 21 days, they're gone. Once you activate an antigen-presenting cell, it's gone after 21 days. That makes sense, because if you have a virus infection and you turn on antigen-presenting cells to the virus and you clear the virus, that immune response can stop. You don't want it to continue forever. So the activated APCs die. If you want that response to continue, you have to reactivate that response. You need to keep boosting and boosting and boosting until the tumor is totally gone. And people aren't doing this. They're not doing it with tumor vaccines. They're not doing it with checkpoint inhibitors. They're not doing it with CAR T cells. They are still doing immunotherapy for tumors based on the old model, which gives the idea that once you activate that immune response, it will continue to go until the antigen has disappeared. And what the danger model says is no. So Steve Rosenberg gave a talk about 6 months ago at the Center for Human Immunology at the NIH, where, among other things, he talked about TIL therapy. And he let slip that he had treated 2 patients with melanoma with TILs 3 times. And he cured both of them. Now, cure is not a word that easily passes Rosenberg's lips. So I asked him later why he doesn't do that more often. And his answer was interesting. He said, I only have so many clinical fellows. I only have 24 hours in a day. I only have so much research money. And I only have so many patients that are willing to give their bodies to science. I want something that's going to work in 2 shots. And I say, ain't going to happen. It's not going to happen. You can get lucky. You can get a few patients whose tumors respond rapidly enough or who have a high enough frequency of cells that after 2 shots you get rid of the tumor. But we can see it. Even with the most modern immunotherapy out there, we're not curing more than 20%. And you know what? Bill Coley was doing that in the 1890s, curing 30% of his patients. So we need to change. We need to stop thinking the old way, because what you think influences what you do. We need to understand that the immune system, once turned on, does not stay on forever, or even during the whole time that an antigen is present. The mere presence of an antigen does not give you an immune response. It can give you tolerance. We need to keep boosting. And there's one guy who did that. And this is the last thing I'll talk about. So here's a guy. His name is Maurizio Bendandi. He is the scientific grandson of a guy named Levy at Stanford, who made the first kind of successful tumor vaccine. Levy worked on lymphoma. Lymphoma is a B-cell tumor. And what he did was, so this is Levy now. So he has a lymphoma from a patient. That lymphoma is making antibody. It's a monoclonal tumor, so a monoclonal antibody. He isolates the lymphoma, fuses them, makes hybridomas, and now he has antibody. And he uses that as the tumor antigen. And so what he does is he takes this antibody and attaches it to KLH, this huge million molecular weight protein, works as a nice adjuvant, and he started vaccinating people. So this is individual vaccine for each person, right? And his protocol, at least, was better than most people, which was 5 shots. So, phase 1. Phase 1 clinical trial is a safety trial. You don't expect any results. You just want to know if you're going to kill people. You have people that have failed all treatment. They're going to die in 6 months. And you're asking, are you going to kill them faster than they would die anyway? His phase 1 trial had 6 people in it. 4 of them went into remission. This was unheard of for a phase 1. Right? These people had already failed every treatment out there. 4 of them went into remission. 1 guy died. And at the end of the trial, 1 guy still had minimal residual disease. And I asked the woman who was running that trial, what are you doing with that guy? And she said, we're waiting. And I said, why? And she said, well, clearly we've cleared 99.99999% of his tumor. The remaining cells must either be escapees or immunosuppressive. And I said, no, they're just not antigen-presenting cells. Boost him! And they wouldn't do it. Now, understand this is a clinical trial. This is human stuff. It's not mice. And you have to get approval from all these boards, and you can't change the trial in midstream. You have to finish it the way you started it, and so on. But that was like 18 years ago, and people are still not doing this in mice or people. Boost him. So Levy had a postdoc named Larry Quack, and Larry Quack had a postdoc named Maurizio Bendandi, and Bendandi heard me talk. He's in Pamplona, Spain. He heard me talk, and he decided to try it. And he did a clinical trial using the same Levy vaccine. By the way, those people eventually relapse. Using the same Levy vaccine, and asked, could he make this kind of okay vaccine better by boosting? So each patient was their own control. It's hard to do a trial when each patient is getting a different treatment, right? Each one is getting vaccinated by their own monoclonal antibody as a vaccine. So what is known about lymphoma is that if you have a tumor growing and you treat it and it goes away, maybe it goes away, but maybe it also comes back. And what happens is many of these people relapse after a certain amount of time. And the reason they relapse is that they have some cells that were resistant to the chemotherapy. And it takes a while for those, or somewhat resistant, and it takes a while for those cells to grow out again. And now they get that chemo again. And what's going to happen is they're going to relapse faster. Because now they're starting with a population that's already resistant to the chemo, and now they relapse faster. And the next time, faster, and the next time the treatment doesn't work at all anymore. So what he did is he took these people that had already relapsed once. They treated them again with chemo. They waited 3 months for the immune system to come back, because the chemo also wipes out the immune system. And then they started vaccinating. So, you have a person, they had this tumor, they were treated, tumor went away, they go a certain length of time, tumor comes back, they get treated again, and what you expect is that they will now relapse faster, that this time period will be less. Anyway, they start vaccinating them, and they vaccinate 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 ad nauseum. Actually, that's not quite right, because they vaccinated them a lot in the beginning, and then less, and then less, and then less. And they published their results, in the Journal of the National Cancer Institute. It got a huge write-up, because the results were so amazing. And it has sunk like a stone. So here are the results. Out of 20 people who responded, not everybody responded, some people are already tolerant. Out of 20 people who responded, 18 of them did not relapse by twice their original relapse time, right? Unheard of. Their original relapse time, or by twice the average worldwide relapse time. Most of them hadn't relapsed by the time they published the paper. They were still immunizing them. One guy died, and I wrote to them and said, what happened? And they said, we ran out of vaccine at 2 years, and he died at 2.5. Okay, so what does this say? We need to think differently from how we've been thinking. If you have a treatment that makes a tumor get smaller, not all treatments make tumors get smaller. If you have a treatment that makes a tumor get smaller, don't stop. Just keep boosting. So that's one of 3 things you need to do for a tumor. A, you need to find something that makes it get smaller, like a vaccine or high-intensity ultrasound, which causes damage, or proton therapy, which causes damage. So you need to find something that makes it get smaller. 2, you need to keep doing that. And there's 2 other things I haven't had time to talk about. 3, you need to make sure that the immune response you get is in the right class. It's a destructive class. And 4, you need to make sure the immune response gets to the tumor. And one way to do that is to do damage in the tumor. Because even activated lymphocytes are going to circulate in the blood, and they're not going to leave the blood until they come to a place where the vascular endothelium has become activated. That's where they leave. So if you want them to leave the blood and go into the tumor, you need to do something to damage the tumor to activate the endothelium. All right, I'm out of time. Thank you so much for listening. Okay.
Just one question about tumor immunology. So you talked about treatment of established tumors. But now I think the Burnett hypothesis, the immunosurveillance hypothesis, has been considerably supported through the work of Schreiber. And what do you think about this? Obviously, you don't believe it. But if it's true, what Schreiber describes, it means that a considerable number of tumors are rejected before ever becoming clinically detected. And what causes then this rejection?
So, I am not a fan of the pure immunosurveillance model. However, data are data. There are data. However, there are data, and then there are interpretations, right? So, when I was a graduate student, it wasn't Burnett, actually. It was this guy in Lewis Thomas. We can't give everything to the Australians. Lewis Thomas, who first suggested that the immune system is constantly going around looking for tumor cells and killing them when they find them, and that when a tumor grows out, it's because it's immunosuppressive or something. That was the original immunosurveillance. But then, we got nerd mice, nerd T cells. And except for a lymphoma, a thymoma, in fact, here and there, like a lymphoma here and there, they don't get more tumors than other mice. And then we got rag mice. And if you don't stick methylcalanthine into them, they tend not to get more tumors than other mice either. Yes, occasionally, here and there, you find a paper where they find, a spontaneous tumor. Most of those are, p53 heterozygotes that they make that way. So no, actually, without an adaptive immune system, you don't see this tumor every 6 minutes that, Lewis Thomas was thinking. We get a tumor every 6 minutes and the immune system gets rid of them, and that rare tumor that we see is immunosuppressive. So, no, I don't believe that model. Now, do I think that there are occasionally tumors that the immune system gets rid of? Sure. I get a melanoma. Actually, here's a really good one from dogs. There's a dog. There are two forms of dog Langerhans cell tumors. Bernese Mountain Dogs get one that kills them, and boxers get the same kind. It looks like the same kind of tumor, but it doesn't kill them. The Bernese Mountain Dog Langerhans cell tumor grows and grows and grows and kills the dog, usually puppy. The boxer gets this Langerhans tumor kind of all over as a puppy, and then one of them necroses and disappears. And then another one necroses and disappears. And then all of a sudden, a whole bunch of them necrose and disappear. And that's it. You never see them again. And there's a guy at Cambridge, England, in the ag department, who looked at those two tumors. And here's what he found. The Bernese Mountain Dog type tumor is a very early stage of Langerhans cell that cannot be an APC. The boxer tumor is a later stage of Langerhans cell. And what he's postulating is that what happens is it starts to grow, and then the dog gets a flea bite, or, playing with its litter mates, gets bitten, and you get some damage in that tumor. And now those APCs get activated, and they induce an immune response to themselves. If I had a melanoma, and it's time for me to practice piano, and I happen to bang my hand on the piano as I walk by it, I'm going to cause damage. If I scratch myself, by the way, why do we scratch? Why have we evolved this mechanism? I think it's to activate local APCs. So yes, I think there will be occasional tumors that the immune system gets rid of, but that it's not a common event. That's, a personal hypothesis.
Okay, I think I promise we'll keep discussing later, but because the coffee will get cold, so I suggest that we take a coffee break.
Yeah, we need coffee.
Half an hour, still possibility to ask questions to Polly privately during the coffee break, and welcome back at 5 to 11. Welcome back, everybody. You did a... Where's Polly? Is Polly back? She's still outside? Okay, I can talk 5 minutes and then she will come back. Thank you for starting so good, asking questions all the time. That's really the way it should be. And I think you enjoy it. Okay, just one thing about question. And one thing maybe I forgot to mention is that the workshop is live streamed. So that people, we are a national society, so that people all over the country, actually all over the world, can watch the workshop. Keep asking questions. But just it means that for the questions to be understandable to everybody, and also because of the live streaming. So please wait for the microphone, or if not, it's important that the speaker repeats the question so that it is well understood and well heard. Now, an important part of the workshop, which I also forgot to mention, that we invited quite a few local speakers, outstanding local speakers, and I will have 2 of those before coming back after lunch to our foreign speakers. So the way it is organized, that we'll have now 2 half-an-hour talks with a quarter of an hour of discussion in between. The discussion is not meant to be just between you and the speaker, but it should be really an open discussion. But let's start with the first one. So the second speaker is Øynebråten. She works at the Department of Pathology at the Rikshospitalet here, working on vaccine development. And she very bravely, she was actually the first one to accept to be a speaker after Polly accepted the invitation. And she bravely accepted to talk about immunogenic cell death. Welcome, Inger.
Thank you. Okay, so yeah, as Alex said, I'm going to talk about immunogenic cell death. And first, to make a definition, immunogenic cell death is a type of cell death that leads to an adaptive immune response. And then it will basically be on, the focus will be on T-cell responses. I will show a few data and refer to few data. Those are not own research data, but data from other research groups. So for immunogenic cell death, there are some requirements. Dying cells, they should provide some immunogenic stimuli, and they should provide antigen. So this is how we envisage it. So the dying cell provide immunogenic stimuli and antigen. These dying cells can be taken up by antigen-presenting cells. The immunogenic stimuli can activate the APC to express co-stimulatory molecules. Antigen will be digested and presented as peptide on MHC molecules, and now T cells can be activated. So just a few slides about types of cell death. So initially, cell death was described as natural cell death to maintain homeostasis, and violent cell death which induced inflammation. Later on, this was divided into apoptosis and necrosis, And this was really defined based on morphology of these two types of cell death modalities. So apoptosis, characterized by cell shrinkage, cytoplasmic blebs, formation of apoptotic bodies, and the plasma membrane retain the integrity. Now, this is also a program type of cell death, and it's energy dependent, and it has been supposed to be non-immunogenic. Necrosis, on the other hand, leads to swelling. There is no vesicle formation, the membrane rupture, and there is lysis. And then it has been described as not non-programmed, not energy dependent, and lead to inflammation. So the last years, these number and types of cell death, this has really expanded in the number of names and the nomenclature. And I think it's still, it is a bit unclear. But this is how people may divide it. So you have apoptosis, which is non-immunogenic. Some also now suggest apoptosis to be immunogenic. Necrosis can be divided into several different types of cell death, and it may also be regulated. It's not only apoptosis that might be regulated. Apoptosis is caspase dependent, and also this form of necrosis called pyroptosis. And as I mentioned, apoptosis, an important characteristic is that the plasma membrane retains its integrity, whereas during necrosis there is membrane rupture. When it comes to immunogenic cell death, there has been quite a lot of studies using anthracyclines and also on some type of photodynamic therapy. Now, these anthracyclines, they showed in mice studies and in immune-competent mice to have better effects on tumor than in mice that were not immunocompetent. And this raised some interest in these drugs. So these drugs, they bind to DNA in the nucleus and to mitochondria, DNA in mitochondria, and halts transcription. They lead to generation of free oxygen radicals, and they are supposed to induce immunogenic cell or immunogenic apoptosis. And examples of such drugs are mitoxantrone, doxorubicin, and epirubicin. Then there is this hypericin-based photodynamic therapy, which hypericin is a drug which localizes in the ER and is a photosensitizer. And this treatment is also supposed to induce immunogenic apoptosis. So, first, is the type of cell death important for the immune response? So this is some data or an experiment from a publication in 2016. And here they used as a vaccine killed cancer cells, and they were killed by use of this photodynamic therapy. And they incubated these killed cancer cells together with dendritic cells, injected it intraperitoneally into mice, and 7 to 8 days later, they injected cancer cells, glioma cells, into the brain of these mice. The cancer cells, as I said, they were either treated with this hypericin photodynamic therapy, as indicated here in red, or these cells, they were just frozen and thawed which may be called necrosis, or which would be necrosis, indicated in green. So as you can see, this treatment of the cancer cells with hypericin photodynamic therapy led to much better, or really protected the mice against severe tumor development. So when it comes to, is the type of cell death important for the immune response? I would clearly say yes, and this has been shown in many studies in mice models. So also based on these anthracyclines and or these treatments that I mentioned, one has identified some markers, some hallmarks of immunogenic cell death. One is calreticulin, which is supposed to be an eat-me signal, so it can help phagocytosis of the dying cancer cell, for example. HMGB1, which can release to the environment and which is a damp. And then ATP, also secreted, and which is supposed to be a find me signal, and which may also activate the inflammasome and lead to interleukin-1 release. So I will talk a little bit about calreticulin and HMGB1 more in detail. So calreticulin, that is a chaperone in the ER, so it helps folding proteins in the ER. It also binds to calcium, and it's important for calcium homeostasis. Now, if you treat cells with anthracyclines, calreticulin will translocate to the surface, and it has been shown to be necessary but not sufficient for generation of an anti-tumor immune response in mice models. So this is a drawing of calreticulin on the apoptotic cells to the left. So typically apoptotic cells, they express or translocate phosphatidylserine. In addition, they may expose calreticulin on the surface. Now this can bind to CD91 on the phagocyte, and which will lead to phagocytosis. Then in the normal cell, you may have some calreticulin as well, but don't eat me signals will be present at higher density and will hinder phagocytosis of the live cell. In an immunogenic tumor cell, you will also have calreticulin exposure after, or at least in certain conditions, which can help phagocytosing. Now, so I was telling you that apoptosis is usually non-immunogenic. Now, whether calreticulin in an immunogenic and non-immunogenic apoptosis differs or is associated with different molecules on the surface may be a reason for why the cells handle them differently. So now, how is calreticulin translocated to the cell surface? In a study from 2009, they tested different inhibitors and quite in detail, and found that if you activate or if you treat cancer cells with anthracyclines, you will have ER stress and you will have activation of PERK. I will show you this in the next slides. They also found that proteins involved in apoptosis was really important for this translocation, such as caspase 8, and of course pathways and molecules involved in secretion. So in the endoplasmatic reticulum, we do have sensors that can note and detect when there is a stressful situation. And a stressful situation is if there is a lot of proteins that has not folded properly, for example. So these yellow things there are proteins that have not folded sufficiently. Now, proteins can detect this, and they will bind to them. For example, this big molecule will bind to these unfolded proteins, and this gives a signal to these different sensors in the ER that something is stressful and something is wrong, and they try to go into a modus where they reduce translation and they increase, but at the same time increase expression of, for example, chaperones that can help folding. Also, now if this leads to severe stress, chronic stress over a prolonged time, the cell may be forced into apoptosis via this pathway ATF4 and SHOP. Okay, so that was calreticulin, and a little bit about HMGB1, which is considered a prototypic DAMP, and it's a ligand of TLR2 and TLR4. It's a sensor for cytosolic nucleic acids, and it's present in nucleated cells, and it undergoes post-translational modifications. And these really determine the localization and function of the molecule. So here is a schematic drawing of the molecule, and there are particularly 3 systems that are important for the modification, post-translational modifications, and some nuclear signals that can also be modified. So these nuclear localization signals, they may be acetylated, and these cysteines, they may be present in a reduced or an oxidized form. So now, if the nuclear localization signals are acetylated, the protein will translocate from the nucleus and into the cytosol. And here it may activate the inflammasome. Now, HMGB1, it may also be secreted, and that could be an active secretion, or it may be passively secreted. So, for example, during necrosis, HMGB1 may be passively released, whereas in other situations, it will be secreted actively. And as secreted molecule, it may have a tolerogenic function. It may induce migration of immune cells, and it can induce production of cytokines. So this is just to show you the different status of the redox situation of the cysteine residues and their different functions that this may lead to. So in a reduced state, which would be the case when the cell is not in a stressed situation, the HMGB1 can bind to chemokine receptors or to RAG and induce cell migration. Now, if you have a formation of a disulfide bridge, the molecule has become a ligand for TLR4 and is able to induce cytokine production. And the most oxidized form is supposed to be tolerogenic. Okay, so there has been many studies on these different immunogenic immunogenic cell death-derived molecules. This is from the same article and study that I just recently showed you, and here they immunized mice in the same way, and in addition they treated the mice with antibodies towards these different immunogenic cell molecules. This is the best response. And this is the mice that received the cancer cells treated again with hyperzine and photodynamic therapy. They showed good survival after injection of the glioma cancer cells. This is the control that didn't receive any vaccine. This blue line here indicates those that received the treatment together with anti-calreticulin. No significant difference, but still a tendency. Now, apirase was used to degrade ATP. So ATP would not be present in this vaccine, and also a tendency, but not significant. And then they used an inhibitor of this reactive oxygen species that is developed because of the treatment, and now they observed a significant reduction in tumor protection and survival. And also when they used an antibody towards HMGB1. And similarly, if they used antibodies towards all these different molecules, they observed a reduced survival. So based on these and other studies, DAMPs are important for development of an anti-tumor immune response. We looked at ATP and calreticulin and HMGB1, and from these data, HMGB1 appears to be the most important one. For me, this makes some sense in a way, because although many of these DAMPs that people list in articles can alert the immune system, I'm not so sure that all of them are really able to activate dendritic cells, for example, which HMGB1 may have the potential to do. Okay, so in summary, the type of cell that is important for generation of anti-tumor T-cell responses in cancer models in mice, and drug treatment can lead to immunogenic cell death based on exposure of HMGB1, ATP, and calreticulin. And based on these treatments, it seems that ER stress sensors and redox status is important, and downstream signaling pathways. So now, immunogenic cell death without drug treatment, does it occur? Does it occur at physiological conditions and in cancer? So in cancer, we know that there is ER stress. We know that there might be, for example, unfolded proteins. There might be a load of reactive oxygen species, both because of hypoxia, nutrient deficiency, and maybe ER stress could be a possible mediator of immunogenic cell death in cancer, and which could contribute to activate APCs. Okay, thank you.
Thanks a lot, Inger. Very nice. Questions or comments? Colin.
I'm just wondering if you could comment on, I guess, the question of whether the damps are changing second signals, or are they also changing the level of antigen presentation itself? So is it really controlling signal 1 or signal 2? Do these experiments look at the absolute level of presentation on MHC of the relevant antigen?
No, so I haven't. I cannot think of any. So these experiments or this study didn't. And right now I cannot remember any studies looking into whether it increased antigen presentation, for example. But I'm sure somebody have done that. Yeah, for sure. And yeah, that I cannot answer you.
Thank you.
Because what would be your idea, Colin, that the dumps would somehow help antigen presentation? What would be the mechanism?
It's needed in order to understand whether the damps are acting as damps or are they changing signal 1 antigen presentation. So if we want to be convinced about the danger hypothesis, then we need to know it's not changing signal 1.
It could do both, said Polly. Microphone, yeah. Wait. Yes.
You'd want to know if it was, so first it could do both. I mean, I can imagine that a damp, a received damp, would change the amount of MHC on an antigen-presenting cell, right? Because the antigen-presenting cell is getting activated. And one of the things that we know they do when they get activated, for example, is flip MHC class 2 from underneath the membrane up very quickly, within minutes. The other thing is we'd want to know, I guess if we wanted to know, whether it was going to change antigen presentation on the presenting cell and or on the tumor cell itself, right? Because if you're going to start injecting these things into tumors, for example, right, it could be doing both. It could be stressing a cell, which would then cause it itself to upregulate MHC, or it could be activating the APC, which would also cause it to upregulate MHC. And I don't see upregulating signal 1 as necessarily mutually exclusive with upregulating co-stimulation or downregulating co-inhibition.
Any more question? Peter, oh, there. Sorry to stop.
So perhaps a related question is a cell undergoing apoptosis inactivating T cells, which would mean it would have to present? And the other question is, could you tolerate the idea that an apoptotic cell is slightly immunogenic and that expansion after the primary decision has been made are controlled by whether it's been apoptotic or necrotic death? So there's sort of two related questions.
So the second question first. So you mean, did I understand you correctly? If you suggest or ask whether the type of cell death play a role after phagocytosis or no?
Yes, after the initial activation, so that it sort of affects the amplification of the response rather than being central in the decision process. You're allowed to be ignorant too.
could of course be but wouldn't you then, if you, then you would expect a immune response in both
Yeah,
conditions,
a small response.
Yeah, a small response.
With apoptotic. I mean, after all, there are a lot of things which are non-cellular, which can induce immune responses without possibly necrosis occurring. We might get into that later.
Yeah, but it may be a possibility, of course.
Sure.
So is that your idea, Peter? Peter, just to formulate clearly your thought, is your idea that the danger signals, so the dumps, are not important for the start of the immune response, but they are in the part of an amplification loop, like in the inflammation?
That's almost what I'm saying. What I'm saying is that can occur, and that you may be able to get initiation without danger, but that doesn't mean that danger can't ever influence initiation. So it's a sort of rather perhaps complicated view. Sorry.
I think it cannot,
Reply by Pauline.
So I know of at least one example where you can get an immune response without danger. And this is why I'm always very careful to say that without DAMPs, you don't get a primary, right? Because if you have antibody-antigen complexes, those can cause aggregation of FC receptors on resting dendritic cells and activate them. So you could certainly get a secondary without danger. And, it makes a lot of sense, actually, that you would want that to happen, because if you've already made antibody to something that has caused damage and given you a primary response, then maybe you don't want to wait until it's done enough damage the second time to get a secondary response. If you already have antibody to something, that's kind of a memory that something has been associated with danger in the past. And so if you can make antigen-antibody complexes early in the infection, before you get enough damage and activation and so on, you could start a secondary without damage. And I think, I have no problem with that. I think that that's actually a really good evolutionary idea.
Okay, back to Peter. And I think Marit had a question after.
So just a couple of points about that. I mean, there is another side to the coin. If you start making a response, and then you don't need control of it through danger, subsequent to initiating it, it means once you've started autoimmunity, it'll go on without any control. And the other thing is that Polly and I have had a discussion before in backwards and forwards in a printed form, where I argued that there are situations where you've got no antibody essentially, and without adjuvant you can get an immune response to red cells in mice. So that's an example where that argument would be weak, that you need antibody to provide a signal for...
I'm not saying you need it. I'm saying that it's
a way of, yeah, okay, sure, I agree.
Yeah, don't put words in my mouth.
Okay, I could just mention for those that are not completely deep in the field that so Peter Brecht has a model where the initiation of the the adaptive immune response is not depending on danger, obviously, and he will present that in details this afternoon. So don't miss Peter's talk.
So just for bit information, the antigen-antibody complex could activate DCs, but what does it mean? It activates DC to upregulate more peptide MHC complexes, or also the second co-stimulatory signals as well?
Again,
Because
I really think that we should hold some of this until Colin talks about, I hope he's going to talk about, co-inhibit and co-inhibition. Because I really
So Colin, will you talk about co-inhibition this afternoon? Yes, he will. Don't miss Colin's talks this afternoon. Okay, any more? There, question, Dennis. Wait,
Yeah.
do you have a microphone? Yes, very good.
Yeah. Inger, I just have a question to you about this HMB protein. How important is endogenous DNA for inducing of these immune responses? Would it be logical to suggest what's release of dump without DNA, endogenous DNA? It would be tolerogenic, and when it's bind...
Dennis, sorry, can you
Yeah.
I'm not
Yeah,
able
sorry.
to hear you.
Would it be, I'm interested in how important is endogenous DNA in the context of inducing immune responses by dump, and particularly by this HBM protein. Would it be logical to suggest that when it's dump bound to endogenous protein, it's much more kind of active to activate toll-like receptor, while it's when it's not bound, it will not activate at such degree, and it will not induce immunogenic response, but it will induce tolerogenic response. I think it's possible to check by injecting recombinant protein without DNA. So what do you think, how it's discussed here? Is it clear? I mean, because when we have a damage of its cells, this protein is released together with DNA. Yeah? If you will just get some protein without DNA, will it induce in the same extent immunogenic response or not?
So you mean HMGB1, for example, with or without DNA?
DNA, yes.
What you're asking. Yeah, so that I am not I do not know this data that well, that I do not answer. But it's not. My understanding is that it's not necessary with DNA. HMGB1 is sufficient without DNA.
Why do we maybe take another question? Is the next speaker in the room? Einar? Yes, he's there. Very good. Maybe you can come down and take the microphone. Or maybe you can take... Is there any more question on immunogenic cell death for the time being? Oh, there is a present for the speaker. She got the present. Thank you again, Inger. Very nice. Now we invited a surgeon with a background in immunology because we wanted to hear more about transplantation, which you've heard already. You've heard Polly's viewpoint, so we're very happy to have you here. Einar Martin. Does the microphone work? Say something. Einar Martin Aandahl is also working at Oslo University Hospital, doing transplantation and immunology research. Welcome.
Okay, thank you for the invitation. So I'm here both as a surgeon and as an immunologist. So I'm working at the transplantation section here at the Rikshospitalet, and what I'm going to talk about is both the surgery and the immunology. And the first part there is I hope that we are doing a good enough job with the surgery. I guess you are hoping that as well. And the real challenge in the field is how to make the organs work better and last longer. And just to take the start, which is the end of the immune response, and this is really the ultimate immune challenge. About 0.1 and 10% of the individual's T-cell repertoire react with alloantigens during the or after the transplantation. And that is compared to 1 in 100,000 for a peptide antigen. And the other thing with the transplant is that it doesn't go away. So it's a continuous immune challenge. And what I'm going to talk about are the processes leading up to rejection. And I guess all of you can see that this is not a physiological place to be for an organ. It's not anatomically and it's not physiologically. And this is actually how we preserve or transport the livers, the lungs, and the heart. It's the liver here. And it's a lot of things that end up with the organ function and the organ survival in the end. It's the physiological processes prior to and during cerebral herniation. About 90% of the donors are dying in a cerebral death. About 10% is what's called circulatory death. I'm not going to talk about that. But most of the patients are dying during a cerebral herniation process. And the next step in the sequence is a warm and cold ischemia. After that, it's a reperfusion of the organ, which ultimately triggers the innate immunity and the adaptive immune response. And in the end, drug toxicity, disease recurrence, and infections also play an important part for the outcome. But we are not perfect either. So a lot of things can go wrong during the surgery, both the organ retrieval and the actual transplantation. There will always be a contusion of the organ. We are handling it with our hands, and it can be organ damage. We can have decapsulation of the kidney. It could be decapsulation of the pancreas, which will give rise to exocrine leakage, abscesses, infections, and a lot of trouble. From the liver, it can be bile leakage. We do some split liver transplants of liver, where we split the liver to give one half to a kid and the other half to an adult patient. Then it will be leakage from the resection surface. We can have hematomas, infections, fluid accumulation, a lot of problems surgically, both during and after the transplantation. I'll just go on. If you can stop me whenever you want. Okay? Who said stop? Okay. Yeah, I will just go on to the next part now, which is immunology. Okay? And this is, we like to look at these curves because the results are getting better and better each year. And this, on the left part, it's the graft survival during the 80s and 90s, and to the right, it's from 2000 to 2015. And the graft survival for the kidneys are excellent. It's the 5-year survival is a little bit less than 90%. Heart is about 80%, and liver is about 75-80%.
As a quick look, it looks like the long-term slopes are the same, but it's the initial rejection which is different.
I'll come to that later.
Okay, good.
Yeah. And what I showed here is the overall effect of all factors, all factored in. What's important as well, which I'll just go on briefly, is the effect of immune suppression and the development of new treatment strategies. And here is the here is the percentage of acute rejections in the different areas. And you can see here it's acute rejection. All this is rejections within 12 months is called acute rejections. And it's less acute within 0 to 6 to 12 months, and it's also less late rejections. That is mostly due to immunosuppressive treatment. And this is the sequence of events from the whole transplantation sequence. To the left, it starts with patient treatment. So this is before a patient is becoming a donor. So this is during this process. It can be organ injuries from the pathophysiological process itself. It can be due to treatment. These patients may have resuscitation. They can have a period of anoxia. About 25% of the donors have died due to anoxia. It could be cerebral edema and infections. And it's a really dramatic change from where we have a patient to where we have a donor. And the donor preservation time is usually measured in hours. That's the time it takes for us to come to the hospital and retrieve the organs. The organ retrieval is about 3 to 4 hours. Then it will be the surgical trauma. It will be the warm and cold ischemia time which starts, and then it will be the cold ischemia time during transplantation. In Norway, we have really short ischemia times. Usually we have the livers in transplanted within 9 hours, and all the kidneys almost are in within 20 hours, which is really short in international terms. The reperfusion is within seconds. And then we have the long period afterwards. It's a post-transplantation time, which is in years, where we can experience acute and chronic infections, acute and chronic rejection, drug toxicity, and disease recurrence. For example, in liver transplantation, which the most common indication in Norway is primary sclerosis, PSC, primary sclerosis in the liver, and the recurrence rate is between 15 to 50%. So this is the process. I'll just show quickly. This is the herniation process, which ends in the cerebral tamponade with complete cessation of cerebral circulation. This is in Norway. 50% of the donors have intracranial bleedings, and about 22% have anoxia of different causes. Traumas is a really small percentage, less than 9%. One of the challenges is that the patient treatment prior to becoming a donor is the opposite of what we want for organ preservation. And the brain death impairs the organ quality through a lot of different processes. And what we know is that, especially in kidneys, where we have both living donors and deceased donors, is that the living donors, the living kidneys, are working much better than the deceased ones. And that's due to the brain death process, and it's also due to the longer ischemia time, the warm ischemia time. But during the brain death process, a lot of physiological processes which end up with increased risk of rejection starts. And the last 3 points there is what's going on. It's a hemodynamic instability, it's hormonal changes, and it's ultimately systemic inflammatory response syndrome. All these processes hampers and reduce organ quality. And the hemodynamic instability, I don't go through all the details here. You can stop me if you want. But the end here is in red. It's huge changes in the hemodynamics. It's hypertension, hypotension. It fluctuates, and it ends up with cardiovascular collapse. And the results for the organ is insufficient perfusion, leading to anaerobic metabolism and acidosis. The hormonal changes is mainly due to cessation of the hypothalamic pituitary axis. And in terms of immunology, the last part is the most important. It's decreased cortisol levels, which promote inflammatory and immunological activation. These patients, the first symptom of the hormonal changes is diabetes insipidus, but this cortisol effect is really important. ultimately, these patients have what's called a cytokine storm syndrome, which is a symptom in the last part of the cerebral herniation process. And during this process, it's a huge release of pro-inflammatory cytokines: IL-1, IL-6, TNF-alpha, interferon gamma. It's HLA upregulation in the organs, which increase the immunogenicity of the organs. It's upregulation of adhesion proteins in the endothelium. The process of apoptosis and necrosis start. Complement activation, coagulation in the organ. We try to flush out all blood to prevent these effects, but it will always be something left. And these processes occurs in a normotension situation, but a lot of these donors have periods of low blood pressure, and this really aggravates the cytokine storm syndrome. I briefly said that it's a difference between living donors and deceased donors, and this is due to the processes I talked about. And actually, if you look in large materials, it's so that a living donor about 80 years old do better than a deceased donor in the 20s. So it's a huge difference between this. So the living donors do significantly better compared to high quality and better matched deceased donors. And this effect is mainly due to acute rejection rates and chronic rejection, which really shows the consequence of the early effects on long-term results. So if we start with the cold ischemia time, which is after the organ retrieval, while we have the organs in plastic bags, or in the kidneys we have in plastic boxes. And during the transportation, in Norway it's about, the longest is in Tromsø, which is about 2 hours plane flight. And then it's the time taking the organs out. If it's a liver, we take the liver out. We put the liver in. It takes about 2 hours. So the time is about 9 to 10 hours before the liver is in. And during this cold ischemia time, that's the hypoxia time. Then it's a production of oxygen radicals. There is a anaerobic metabolism with lactate production. And it goes to process I'll talk about now. But the direct effect of the cold ischemia time is increased risk of what's called delayed graft function. In kidneys, delayed graft function is the need for dialysis within the first days after transplantation. And the incidence is about 15 to 50%, depending on the materials. And what we know is that what induces this delayed graft function is donor age and duration of the cold ischemia time. What's been shown now is it's a direct effect of the cold ischemia time, the time of ischemia, with the risk for acute rejection. I'll just take it a bit faster, but what's happened during the ischemia time is that we have the metabolic stress and ultimately necrosis of cells within the organ. And it's the release of the DAMPs and the ROS and all the different metabolites or signaling proteins and danger activation part leads to immune activation. Immune activation gives further rise to cytokines and proteases, which drives the necrosis in the organ. At the same time, it's a systemic effect with activation of peripheral lymphocytes, monocytes, and so on, which also sustain the inflammatory effects. Looking at the patient data, it's so that each additional hour of cold ischemia time increases the risk of graft failure and graft rejection. Ischemia times of 30 hours, we never have 30 hours in Norway. But in Europe and the US, where the transportation distances and times are longer, they can have ischemia time up to 36 hours. But a 30-hour cold ischemia time have a risk for graft failure near 40% higher than 6 hours of ischemia time. And the same is with the risk for death of the patient. We also know that the older the organs are, the higher the risk for graft loss, acute and chronic rejection. The two curves here is donor age less and more than 55 years old. Then it's a perfusion injury, which leads to microvascular injury, fluid filtration, edema in the tissues. Endothelial secretes inflammatory mediators, and the endothelium loses its integrity, apoptosis, necrosis, and ultimately delayed graft function. These are the different effects of the reperfusion, ischemia reperfusion injury: vascular leakage, apoptosis, necrosis, transcriptional reprogramming, and autoimmunity, stimulation of recurrence of autoimmunity, and ultimately activation of innate and adaptive immune responses. At the cellular level, the effects of cold ischemia time and the reperfusion injury is cytoskeletal disruption of the histological microarchitecture within the tissues. The ATP exhaustion leads to conversion and changes in the actin, cytoskeleton constituents, and ultimately it ends up with upregulation of TLR4, which is really, it's a lot of focus on toll-like receptor 4 in the transplantation field, and activation of the immune and tissue-specific cells. And obviously, release of DAMPs. This is the whole context that we're working with in transplantation. This is, on the left here is a graft, on the right is a host. In the graft, we have the cold ischemia time, reperfusion injury, cell lysis, release of DAMPs, and within the tissue it will be an inflammatory response, activating dendritic cells, macrophages, and adhesion molecules in the endothelium. And the release of cytokines from the tissue-resident inflammatory cells leads to activation within the host. And this is the important part. Here, the really adaptive immunity starts. The activation of the inflammatory response in the host further increase, or these cells will infiltrate, go into the graft tissue, further stimulate cytokine production and chemokines, leading to activation of the adaptive immune response, which can go both ways, which I will show later. Toll-like receptor 4, which is which is a big focus within the transplant field. It signals can signal through different adaptive proteins. MID88 is one of them, and it also ends up with nuclear translocation and activation of NF-kappa B and cytokine production. DAMPs and toll-like receptors have been shown to be important in all different human transplantations, in kidneys, lung, liver, heart, and also pancreas. And as showed in the previous presentation, HMGB1 is one of the important DAMPs activating toll-like receptors. This is from a mouse model with looking at TLR4 activation and expression in both infiltrating immune cells and also in tubular cells in the kidneys after reperfusion of the organs. And the upregulation of TLR4 is rapid in the infiltrating cells, but it's also sustained for days after transplantation in the tissue resident cells. Here, the tubular cells in the kidneys. So TLR4 is expressed both by inflammatory cells and the tissue cells. This is a mice experiment with knockout mice, with TLR4 knockout mice and mid-88 knockout mice. And here it's a serum creatinine on the y-axis, and the knockouts have lower serum creatinine after reperfusion compared to the wild types, which indicates that both TLR4 and mid-88 mediate some of the tissue injury. This is a really interesting paper. It's an old paper from 2005, but here they looked at 238 kidney recipients, where 27 of these patients had a loss of function, TLR4 polymorphism, and they looked at the outcome in terms of acute rejection, infections, opportunistic infections, and ultimately graft function. And it supports a hypothesis that TLR4 is important for the acute rejection, but it's on the expense of infections. So these patients experience more severe bacterial infections and also more opportunistic infections. So it's not as easy as to just to manipulate the TLR4 function. I don't know. Maybe it's also contributing. Yeah. I don't know. Does anyone know? And the last part of this sequence of events is the activation of adaptive immunity and ultimately organ rejection. And going back here, we are now in the end of the inflammatory sequence, and this is what's in the textbooks, all textbooks on transplantation immunology. Here is the direct allospecificity in this part, and then it's a late phase, post-transplantation phase here. And in the first phase, it's a donor dendritic cells that will go to the lymph nodes and present alloantigens. So it will be both donor-derived dendritic cells or antigen-presenting cells presenting foreign antigens, alloantigens. In the late phase, it will be the recipient-derived recipient's dendritic or antigen-presenting cells migrating to the allograft and taking up alloantigens and then going back to the lymph nodes presenting the alloantigens in the context of self-MHC on self-dendritic cells. But this is not the whole story. The complement cascade is also important in terms of outcome in organ transplantation. And in kidneys, we actually use a C5a monoclonal antibody in some settings. And what's important is that during brain death, it's an upregulation of complement activation within the organ, and most of the complement components like C3 is derived from the kidney itself. 10 minutes. But I'm almost done, so we have more time for discussion. Okay, so this has been a controversial point. It's whether the cold ischemia time have a direct effect on rejection. And this is from a paper from 2008, where they looked at cold ischemia time longer or shorter than 24 hours, which is a long time. Actually, when you look at huge materials, there is a cutoff on 20 hours. And it's really a big difference, shorter than 20 hours and longer than 20 hours. And that's our experience here as well. So we always try to have shorter than 20 hours. But here they have 24 hours. In some papers, they even have 36 hours, which is really, really long. But here they show the effect of cold ischemia time shorter or longer than 24 hours. But what's really interesting is that when they stratify for acute rejection, the effect of cold ischemia time disappears, which indicates that the effect of cold ischemia time is due to rejection. So that's a really interesting observation. And then we end up with the conclusion that the consequence of the cold ischemia time, the reperfusion injury, is due to increased rejection. And just to show in the end here, what is the effect of acute and late rejection? And this paper from 2015, they have really dramatic findings. But here it's on the x-axis here, it's the survival curve for the patients, and it's really decreasing with acute rejection and late infections. And looking at graft survival, it's even worse. This is without rejection. This is with early and late rejection. So rejection has a really important consequence for kidney allografts. It does not have that dramatic effect on liver grafts, and that's due to different factors. It may be that the liver is more tolerogenic than the kidneys. The kidneys will accumulate damage or injuries, fibrosis during the whole continuously during time, while the liver has a regenerative capacity. But for kidneys, the effect of rejection is really detrimental on graft and patient survival. And then, what are the therapeutic opportunities? Inhibition of toll-like receptors. I showed you that may be effective on rejection, but it has the cost of risk for infections. Complement inhibition, that's up and coming. C5-8 inhibitors are in clinical trials and also in clinics in some settings. Regulatory T cells will be important. And then other immunoregulatory mechanisms, CTLA-4, we're using CTLA-4 agonists in the clinic, belatacept. PD-1 ligand may be in the future. But what we're using now is ex vivo perfusion machines. And that's a mechanical installation to reduce the effect of oxygen radicals, cytokines accumulating in the graft, washing out lactate. And the machine looks like that. We are using it at this hospital. We use it when the donors are more than 70 years old, and we put the latest kidney in the machine. That will be when we do the organ retrieval, we have 2 kidneys, and the kidneys which will be transplanted last will be on the machine. So we have 1 kidney on the machine. It will be the one which will be transplanted last. It's mostly used for kidneys. There are some machines now for liver perfusion as well, but we only use for kidneys. And the effect is quite good. This is with the machine perfusion. This is cold storage, what's called static cold storage, which is in the plastic bags and the boxes, and graft survival on the y-axis. What we think is the effect is preservation of the endothelium, reduced effect of the reperfusion injury with oxygen radical cytokines, so on. And what's also is an up-and-coming question is whether we should use perfusion machines which introduce oxygen or non-oxygen perfusion. We use non-oxygenated perfusion here. And it appears that it's better with oxygenated perfusion fluid. Okay, so that was the last.
Thank you very much, Einar. That was great. Questions? I'm sure we can probably have to. There's a question in the back there.
Yes, very nice presentation. I was just wondering what would happen to your patients if you would subject them to cold. So by that I mean periodical cold exposure, like let's say, I don't know, maybe not swimming in the lake, but something to maybe disconnect those 4 steps so that you don't get activation of adaptive immunity.
I don't have a question. I have an answer on that. I don't think anyone has. But what has been discussed a lot is is it beneficial with having the donor cold or not? Was that your question, or was it the recipient?
I mean, the basis or the premise of this question is that I think there was either paper in Nature or Cell.
Recipient, yeah.
Huh?
You meant the recipient should be.
Yes, the recipient
cooled
after
down.
the transplant. Because I was just wondering whether or not, whenever we get sick, right? Usually we have an increased temperature, right? I think there was one paper showing that the actual immune response is much higher when you do not dampen the body temperature. But if you do, then the time that your body takes to combat the infection is longer. So I was thinking in the opposite way. If you would,
I don't have an answer on that. I'm not sure whether it would go through any ethical board.
Yeah, that would be quite difficult to convince someone that the patient.
What has been done a lot is the effect of hypothermia on the donor during the organ retrieval, and it's not really clear the effect there. It's hypothetically it would be nice to have the donor cold, right? Because you will stop a lot of these effects. But reducing the temperature also have an effect on the hemodynamics of the donor and risk for hypotension, which is not beneficial for the organ. So it's not a clear answer on that. So when we're doing the surgery, we adjust the temperature to the recipient and to the surgeon, so that we're not sweating.
Yeah, thank you.
Yes, Eric Torsby in the middle.
Thank you. Allow a survivor from the early days of transplantation immunology to add 1 point on the therapeutic opportunities. And I still would argue for good HLA matching, in particular for long-term results after grafting. That's just a comment.
Yeah, and that's a good point. And we do HLA matching on kidneys. We don't do it on livers, but we do it on kidneys. And what we really are aware of is doing good HLA matching on young recipients because of risk for immunization for later transplantations.
Yes.
So, and the kidneys, for those who don't know it, the kidneys last about 10 to 15 years. So if you have a recipient which is 30 years old, the patient will likely be transplanted 2 or 3 times. And the HLA matching is really important for those patients to avoid
I'm very
immunization.
happy to hear that. Now, my question, coming back to tissue damage and the dangerous signals of poly, what about chronic rejection? I mean, antibody-mediated chronic rejection. Do you see that more often using deceased donors than in living-related donors?
There are 2 answers to that. The impact of long-term antibody-mediated rejection is being recognized in the latest years now. On kidneys, the chronic allograft nephropathy has been attributed to calcineurin inhibitors, but it's evident it's more likely now, or it's been recognized that it's due to subclinical antibody-mediated rejection. But the answer to your question is that we don't know. So the effects of cold ischemia time, the DAMPs and so on, has been shown on acute and late acute rejection, not long-term.
My question actually relates to would excessive tissue damage, many dangerous signals lead to long-term maintenance of activation and thus also more chronic rejection using deceased donors? But you don't know.
Probably.
No, probably, yes.
And immune activation due to infections and so on also contribute.
Can I ask you myself, Einar, using these perfusion machines, I think you call them, to preserve the organ before transplantation, do you think there would be a possibility to use this machine to remove the dendritic cells from the organ, which according to the danger model would make
you mean
danger-free?
Flushing out the dendritic cells and
or
inflammatory
actually
cells?
they're supposed to come out by themselves. I mean, that's their job to get out of
maybe.
through the lymph. So, is anyone working on this approach?
There are some people here working on perfusion machines, and I think that maybe warm perfusion and perfusion with blood makes a more physiological situation and may also be beneficial for having immune cells migrating from the tissue into the circulation and being washed out.
Right.
But I know there are some people here working on that. Maybe they won't answer.
Isn't it your prediction, Polly, that if you remove the disease, then the graft should be accepted?
Well, if you remove the disease, you're now stuck with the host disease, right? So certainly the acute rejection will go down, but I don't think it'll go away.
Okay.
But does somebody who works on these machines want to answer that question? No? Yes?
That's the question.
Well, Anna Martin, you were also involved in that. So we are working with the machines, but we have, until today, not evaluated the dendritic cells in the ileate, which is coming out of the kidneys. We are mainly working on to inhibit the innate immune reaction on the damage. And we'll present this a little bit about that tomorrow.
Very good. Can I ask another question, Einar? Polly mentioned in her talk that one problem with cyclosporin was that it was blocking signal 1 and then preventing tolerance after a certain time in the whole. So the idea would be that you could remove immunosuppression if tolerance had been induced by the graft being in the host.
Yeah.
So, but I think, what is the situation nowadays? Are you still using cyclosporin to treat the patients? Are the drugs on the market that are not blocking signal 1?
Now we usually use tacrolimus. but it's the same. It's a
The same.
calcineurin inhibitor.
Okay.
But some of the patients that's been transplanted years ago are still using cyclosporine. But in terms of tolerance, it's quite interesting with liver, because liver have a higher tolerogenic effect. And what we know from compliance studies is that about 20% of the patients will be tolerant to the graft, but we don't know who they are. But that's from compliance studies, that people haven't followed the treatment and so on, and they're still not rejecting the organ. But that's liver, not kidneys.
But didn't Roy Kahn show that in rats a long time ago?
Sorry?
So Sir Roy Kahn, C-A-L-N-E, a British liver transplanter, long time before you were born.
Yeah, sure, I was born.
Showed in rats. So that if you transplant a liver in some strains of rats, it's accepted. And you can later transplant a heart from the same donor. But if you transplant that liver under the cover of cyclosporin, now the liver can't induce tolerance that will give the heart acceptance. So he already said, I think in the 70s, that cyclosporin was a bad idea as far as tolerance induction.
But it's the terms, the concept of rejection is really complicated, because when we do pancreas transplantation, it's usually combined kidney pancreas, because the patients that we transplant is diabetic patients with nephropathy. And it appears that you can have rejection in one organ, but not the other, which is difficult to understand.
No.
You don't think so?
I don't think it's difficult. Are you going to talk about split tolerance? No. So different organs have different antigens. And probably also different susceptibilities to certain classes of immunity. And so you could have tolerance of one organ. So, for example, again, mice and rats, you can make an animal tolerant of bone marrow, fully allogeneic bone marrow, and yet it still rejects skin. And it's because the skin has antigens the bone marrow doesn't have. So I can imagine that you could be tolerant of kidney and not of pancreas, or you could become tolerant of pancreas and not of kidney. But the other reason it could also be because some tissues are more susceptible to, CD8 killing and TNF and so on, and other tissues less susceptible to that.
But still, it's the HLA effect, which is really strong antigen.
Sure. Yeah, but HLA, so my very first paper was the suggestion that the reason we have such a high frequency of cells to HLA differences is that they are not to the HLA itself, but to HLA in combination with 1,000 other, now we know, peptides, right? And so again, you can imagine that the peptides that are expressed by a skin cell in HLA are not all the same as the peptides expressed by a kidney cell or a bone marrow cell in HLA. And that also has been shown in mouse transplantation studies.
And it also appears that the amount of tissue that you transplant is important. If you go back to this with combined pancreas-kidney, which is called SPK, simultaneous pancreas kidney transplantation, it's less rejection of the pancreas than with pancreas alone.
And isn't the same also true with liver and heart?
I don't know.
So I have a couple questions, comments. I noticed that in your list of potential treatment...
Exam time, Einar.
Sorry?
Your exam time, yeah. Questions by Pauline.
Sure.
So, your exam time, you're too funny. So, in your list of potential treatments, you didn't mention co-stimulation blockade. And yet we have Alan Kirk's work showing that, anti-CD40 ligand, for example.
Sorry,
anti-CD40 ligand in monkey kidney transplants can be given for about 5 months and then stop, and the monkeys don't reject. So why isn't that on your list of potential treatments?
These are the main strategies used in the human setting now. I'm not sure whether CD40 perturbation is in any clinical trials? Do you know?
They started anti-CD40 ligand trials.
Excuse me, Polly, could you explain just a little bit better? So what is the idea of blocking CD40? Is it on dendritic cells, on B cells?
B cell activation.
So the CD40 ligand is on T cells.
Yes.
And CD40 is on the dendritic cells. And they used anti-CD40 ligand to prevent the CD40 ligand CD40 interaction that superactivates dendritic cells. But the problem was when they went into clinical trials, there had been some other clinical trials using anti-CD40 ligand in like atherosclerosis. And they found that CD40 ligand is also on platelets. And then what happened when they gave the anti-CD40 ligand, they got thrombosis. They clotted the platelets. And so the transplantation studies were stopped because of the atherosclerosis results. But another reason they didn't go into clinical trials, according to Alan Kirk, is that the company that makes anti-CD80 and the company that makes anti-CD86 and the company that makes anti-CD40 ligand is 3 different companies. And they were unwilling to be part of the same clinical trial. Because if something goes wrong with the other company's reagent, you don't want to get sued, right? So, okay, but here's another one. So Walter Land, who used to be the head of experimental surgery in Munich, had a study in kidneys, in human, not monkeys this time, right? Where they had 44 patients that were treated in the normal way, and another 44 patients that were given a single injection of superoxide dismutase, so antioxidant radical, at the time of transplant. And the ones who got the SOD had almost no early acute rejection episodes. And so I wonder why that isn't on your list of potential treatments.
It sounds as it should be.
Sorry?
Yeah, it sounds as it should be.
Yeah, it sounds so. Maybe that's something to go look at. And I had
some really radical strategies for tolerance, which is completely different, is whole body irradiation and then bone marrow transplantation with organ transplantation from the same donor. But that's a different setting.
That's pretty drastic. But
that's drastic.
Giving bone marrow along with kidney, and some immunosuppression might not be a bad idea. And then there's one more. So you mentioned SIRS. And there's a trauma surgeon at Harvard who has shown that this huge cytokine storm that looks like a response to sepsis is a response to mitochondria released from the damaged tissue. And that you can block some of that response with an antibody to the formylmethionine peptide receptor on neutrophils. And so that would be also something that I would think you might want to consider adding to your list of treatments.
In the transplantation setting.
Any final question from the audience? There is. Very good. And then we'll take a lunch break. One more question.
So is there anything known about the role of exogenous factors? For example, you transport the graft in a plastic box. So how safe it is?
How safe the transportation is?
Not transportation, but the material itself, the plastic.
Yeah, that's safe. So actually, the plastic bags that you saw there, we have 3 layers of plastic. We have the first is for the organ with preservation fluid, and then it's another bag with ice and water, and then it's the last one is just protection. But the plastic bags we use are plastic bags that are used during surgery for different conditions as well.
Because, for example, the gold, transporting them in a gold-coated box, for example, gold particles somehow reduces antigen presentation, suppress antigen presentation by disease. So that's what I learned. so transporting them
So
in a coated box might...
the key point in the transportation is the preservation fluid.
Very good. Thank you to all speakers. So now we take a 1 hour break. Please come back at 1:30. Yes.